Hello everyone, today we are going to talk about Alzheimer's disease beyond amyloid-emerging drug targets. This is presented by me and Ted. I will covering the material up to 12 targeted therapies and the remaining session will be presented by my partner. Just to mention, for some parts I will go over the main ideas and keep the details brief so we can stay on time. So alright, let's get started. What is the Alzheimer's disease? Alzheimer's disease is a progressive brain disorder. It usually starts with the short-term memory loss and progressive cognitive decline. It is mainly characterized by amyloid plaques outside the neurons and neurofibrillary tangles inside the neurons. Now, I will explain how amyloid beta is formed using the diagram. So I skip this slide, and the diagram covers all. There are two possible pathways depending on where the first cut happens. On the non-amyloid pathway, the alpha secretase cut inside the amyloid, producing the hemlock fragment, so this pathway is safe. But on the other hand, on the amyloid pathway, beta secretase makes the first cut outside the amyloid beta region here, so the amyloid sequence is preserved. This releases the amyloid beta peptides and they can stick together and forming oligomers. They then aggregate and then eventually plaques and tangles. Now that we have talked about the amyloid, so let's move on to another key protein, which is Tau. Tau is a protein found inside the neurons, and its normal role is to stabilize the microtubules. But in Alzheimer's disease, Tau becomes the hyperphosphorylated. This means too many phosphate groups are added. Because of this, Tau loses its ability to bind to microtubules and starts to stick together and form neurofibrillary tangles inside the neuron. These tangles disrupt the cell function and lead to neuron damage and cell death. And positive feedback cycle loop may occur because like amyloid and tau may reinforce each other and lead to the disease progress. Now that we understand how amyloid forms, and let's look at the treatment that's studied. Some drugs are designed to remove the amyloid plaques directly, for example, aducanumab. These binds to the aggregated amyloid beta and help clear the plaques from the brain. Vaccines are also being studied, which aim to stimulate the body to produce antibodies. So overall, these treatments focus on the reducing amyloid buildup. Besides the antibodies, researchers are also studying the small molecule drugs. In this case, these drugs work by preventing proteins from sticking together, which reduces the aggregation. So instead of removing plaques, they try to stop them from forming in the first place. However, even with these treatments, there are still important limitations. They only provide the minimal slowing of the disease progression, and they do not completely stop the cognitive decline. And moreover, they can cause side effects such as brain swelling. So overall, targeting amyloid alone is not enough. So this is why the researchers are now looking beyond the amyloid to other targets. So this brings us to the important question: what comes after the amyloid? A common phrase that is used is amyloid is the trigger and tau is the bullet. This means the amyloid may start the disease, but tau is more directly responsible for the neuron damage. Cognitive decline becomes more noticeable as tau increases. So this is why the targeting tau is becoming more important in research. However, targeting tau is not simple as we said. Tau exists in many different forms due to the chemical modifications, such as phosphorylation and other changes. Because of this complexity, it is difficult to design drugs that target all forms effectively. This is the overview of the strategies. These include the targeting Tau directly, like Tau aggregation inhibitors, reducing its production, Tau expression inhibitors, modifying its structures here, and restoring its normal function. So this slide gives the big picture, and the next slides will go into more detail. And the first approach is targeting Tau using the immunotherapy and gene-based methods. There are two types. Passive immunotherapy uses antibody directly to Tau and help remove it. And active immunization therapy works by stimulating the immune system to produce its own antibodies against Tau. There are also gene-based approaches. So overall, these methods focus on reducing the amount of harmful Tau in the brain. Another approach is targeting the chemical modifications of Tau. In Alzheimer's disease, Tau is especially hyperphosphorylated, so removing or controlling phosphorus is important because it may help restore the Tau normal function and prevent tangle formation. And there are two types, kinase inhibitors and phosphatase activator that provide comprising phosphorylation control and other methods to reduce or reverse these modifications. The goal is to stop Tau from becoming toxic in the first place. Finally, another strategy focuses on restoring tau normal function. When tau is damaged, microtubules become unstable and transport inside the cell is disrupted. So microtubule stabilizing drugs may help restore this function and improve cell stability. So instead of removing tau, this approach tries to fix the damage it causes.
Okay, let's move on to another important part, which is microglia. Microglia are immune cells in the brain. Their main role is to remove protein aggregate and debris through a process called phagocytosis. In Alzheimer's disease, microglia became less effective at clearing these harmful substances, so researchers are studying ways to restore or enhance microglia activity. However, targeting microglia is not simple. If microglia are overactivated, they can start damaging healthy brain cells. Earlier treatments like anti-inflammatory drugs did not show clear benefits, so this showed that simply suppressing inflammation is not enough. The response needs to be carefully controlled. This brings us to a specific target on microglia called TREM2. TREM2 is a receptor that helps microglia recognize harmful proteins like amyloid plaques. When TREM2 is activated, it can improve the ability of microglia to clear these plaques. However, earlier antibody treatments were not effective. Instead of activated TREM2, the goal is to be removed from the cell surface, which reduces its function. Another important system involved in Alzheimer's disease is the complement system. The complement system is part of the immune system that helps tag cells or structures for removal. However, in Alzheimer's disease, this process became overactive. Too many synapses get tagged and removed. This leads to excessive synapse loss, which contributes to cognitive decline. So, the complement system is now being studied as a therapeutic target to better control microglia activity. Now, let's move on to another important factor, which is ApoE, which is a protein involved in cholesterol transport in the brain. ApoE is also found in amyloid plaques and play a role in both lipid metabolism and plaque biology. There are different forms of ApoE gene. ApoE4 increases the risk of Alzheimer's disease, while ApoE2 is considered protective. Scientists aim to shift ApoE4 toward the protective ApoE2 form. Other strategies focus on regulating cholesterol metabolism or reducing harmful ApoE effects. So overall, targeting ApoE is challenging, but it is an important area of research because of its strong links to Alzheimer's risks. In Alzheimer's disease, brain metabolism changes over time. Conditions like diabetes are known to increase the risk of Alzheimer's disease. Metabolisms and inflammations are closely linked. Some drugs like GLP-1 drugs may reduce inflammation, but they have not shown clear cognitive improvement yet. So, metabolic targeting is still under investigation. So, what does the futures of Alzheimer's treatments look like? Researchers are now moving toward multiple target strategies and combination therapy. This is called synergism, where two treatments work better together than alone. Treatment may also be personalized based on biomarker. For example, biomarkers such as amyloid betas and tau levels in patients' cerebrospinal fluid or brain imaging like PET scan can help identify the stage of the disease and guide personalized treatment. These are the reference used for this presentation. To conclude, Alzheimer's disease involves multiple complex processes, and future treatments will likely use combinations and personalized approaches. Thank you for your time and attention.